Hello students, welcome to Read MedPrep Academy channel. Today in Chromosomal Basis of Inheritance Part 3, we are going to talk about crossing over, mechanism and importance of crossing over, recombination and genetic mapping. What is crossing over? It is a biological process that produces a new combination of genes by interchanging the corresponding segments between the non-sister chromatids of the homologous pairs of chromosome. The term crossing over was coined by Morgan in 1912. It takes place during the packaging stage of the prophase 1 of meiosis. Here in this image, you can see the crossing over during the packaging stage of prophase 1 of meiosis. In the leptotin stage, the prophase begins with the chromosomes starting to condense. In the zygotin stage, the synapses begins with the synaptonemal complex formation. In the packaging stage, the crossing over DNA is exchanged by the non-sister chromatids. In the diplotin stage, the synapsis ends. Chiasma is visible within the bivalent. In the diakinesis stage of prophase 1, the prophase ends and the nuclear membrane disintegrates. Here you can see a close-up view of the crossing over during the packaging stage. The homologous chromosomes come close together and there is chromosomal crossing over the non-sister chromatids, and there are formation of two recombinant chromatids and two non-recombinant chromatids. Usually, crossing over occurs in the germinal cells during gametogenesis. It is called meiotic or germinal crossing over. It has universal occurrence and has greater significance. Here you can see that when there is no crossing over, the result is all the four non-recombinant chromatids are formed. When there is crossing over, there is formation of chiasma between the non-sister chromatids, and the result in the formation of two non-recombinant chromatids and two recombinant chromatids. So there is formation of four chromatids with two recombinant and two non-recombinant chromatids. Rarely, crossing over occurs in the somatic cells during mitosis. This is called somatic or mitotic crossing over. Now, what is the mechanism of crossing over? There are different stages of crossing over. The first stage is synapses, the second stage is tetrad formation, the third stage is crossing over, and the fourth stage is terminalization. What happens in synapses? Intimate pairing between the two homologous chromosomes is initiated during the zygotin stage of the prophase 1 of meiosis 1. Homologous chromosomes are aligned side by side, resulting in a pair of homologous chromosomes called bivalents. Here in this image, you can see the two homologous chromosomes come close together. There are sister chromatids of the same chromosome and the non-sister chromatids of the different chromosome. And there is a formation of bivalent. And the crossing over occurs of the non-sister chromatids, resulting in the formation of the chiasma. This crossing over results in the formation of two recombinant chromatids and two non-recombinant chromatids. What are the different types of synapses that occurs? The pairing phenomenon, which is called synapses, is of three types. Procentric synapses, pairing starts from the middle of the chromosome. Proterminal synapses, pairing starts from the telomeres. Random synapses, pairing may start from anywhere. Here you can see in this image the synaptonemal complex formation, where the sister chromatid 1 and 2 is seen and the sister chromatid 3 and 4 is seen. In between, there is a formation of synaptonemal complex. In the synaptonemal complex, there is combination of recombination nodules, and these nodules are responsible for the crossing over. Now, what is tetrad formation? Each homologous chromosome of a bivalent begin to form two identical sister chromatids, which remain held together by the centromere. At this stage, each bivalent has four chromatids. This stage is called tetrad stage. Here you can see the tetrad formation. Two homologous chromosomes come close together and there is crossing over and the formation of the tetrad. After tetrad formation, crossing over occurs in the packaging stage. This stage is called crossover. The non-sister chromatids of the homologous pair makes a contact at one or more points. These points of contact between the non-sister chromatids of homologous chromosomes are called chiasmata. Singular, it is called chiasma. At the chiasma, the cross-shaped or egg-shaped structures are formed, where breaking and rejoining of two chromatids occurs. This results in reciprocal exchange of equal and corresponding segments between them. Here you can see the formation of chiasma and reciprocal exchange. The two homologous chromosomes come close together. There is a formation of bivalent and tetrad, and there is a crossing over of the non-homologous chromatids, non-sister chromatids of the homologous chromosome, and the chiasma is formed. This results in the formation of two recombinant chromatids and two non-recombinant chromatids. Now, what is terminalization? After crossing over is over, the chiasma starts to move towards the terminal end of the chromatids. This is known as terminalization. As a result, complete separation of the homologous chromosomes occur. To sum up the mechanism of crossing over, you can see in this image, in the prophase 1 of meiosis, the non-sister chromatids of the homologous chromosomes come close together. In the packaging stage of prophase 1, there is a formation of X, or the overlapping of the non-sister chromatids, resulting in the formation of the chiasma. This point of crossing over is called the chiasma, which results in the exchange of genetic material. In metaphase 1, there is slowly separation of the homologous chromosomes. In metaphase 2, the chromosomes are separated. What is the result? Two parental non-recombinant chromatids are formed and two non-parental recombinant chromatids are formed. Now, what is the importance of crossing over? Crossing over occurs in all organisms like bacteria, yeast, higher plants and animals. Its importance is exchange of segments leads to new gene combinations, which plays an important role in evolution. Studies of crossing over reveal that genes are arranged linearly on the chromosomes. Genetic maps are made based on the frequency of crossing over. Crossing over helps to understand the nature and the mechanism of gene action. If a useful new combination is formed, it can be used in plant breeding. Now, what is recombination? Crossing over results in the formation of new combination of characters in an organism. These are called recombinants. In this, segments of DNA are broken and again recombined to produce a new combination of alleles. This process is called recombination. How to calculate the recombination frequency? The percentage of recombination progeny or recombinant progeny in a cross is called recombination frequency. The recombination frequency or crossover frequency, otherwise represented as RF, is calculated by using the following formula. The data is obtained from the alleles in the coupling configuration. RF is equal to total number of recombinants divided by total number of progenies into 100. What is meant by heteroduplex? A double stranded molecule of nucleic acid originated through genetic recombination from different sources is called heteroduplex. Now, what is genetic mapping? We know that genes are present in a linear order along the chromosome, and these genes are present in specific locations, and these are called locus. In the plural, it is called loci. The diagrammatic representation of the position of genes and related distances between the adjacent genes is called genetic mapping. It is directly proportional to the frequency of recombination between them. It is also called as linkage map. The concept of gene mapping was first developed by Morgan student Alfred H. Sturtevant in 1913. It provides clues about where the genes lies on the chromosome. The unit of distance in a genetic map is called a map unit, MU. One map unit is equivalent to 1% of crossing over. One map unit is also called a centimorgan, represented as small c and capital M, in honor of T.H. Morgan. 100 centimorgans is equal to 1 morgan. Now, one example of map unit, for example, the distance between the A and B genes is estimated to be 3.5 map units. It is equal to 3.5 centimorgans or 3.5% or 0.035 recombination frequency between the genes. If you take the distance between B and C, it is around 7.5 map units. So it is equal to 7.5 centimorgans or 7.5% or 0.075 recombination frequency between the genes. If you take the distance between C and D, it is around 5.5 map units. It is equal to 5.5 centimorgans or 5.5% or 0.55 recombination frequency between the genes. So what are the uses of genetic mapping? It is used to determine gene order, identify the locus of a gene and calculate the distances between the genes. They are useful in predicting results of dihybrid and trihybrid crosses. It allows the geneticist to understand the overall complexity of the particular organism. So today in chromosomal basis of inheritance part 3, we discussed about crossing over, the mechanism and importance of crossing over, and what is recombination and what is genetic mapping. Thank you. Kindly subscribe, like, share and comment our channel ReadMedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is ReadMedPrep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you, students.